I want to welcome everyone to our Clinical Trials for Rare Diseases Session 2, Clinical Trials 101, and the Family Perspective. I want to especially welcome our presenters that represent some of the families and doctors that have been involved with clinical trials, either as patients or trial leaders. So I'm hoping you will learn more about what's involved as a patient, if you choose to be part of a clinical trial. Now I'd like to introduce Laura Eskew from ICON that will lead us in tonight's webinar. So Laura, I turn the meeting over to you.
Thank you so much, Bob. Next slide, please. And it is an honor to be here this evening. I'm delighted this is one of my favorite things, to share information about clinical trials with communities. Thank you, each of you, for taking the time to be here and take this information back to your families and to your communities at large. This evening's program is taking place in 3 parts. First, we're going to have a presentation about clinical research and navigating clinical trials, and that will actually be done by me. And following that, we have 3 wonderful individuals who are going to be sharing their experiences or the experiences of their patient communities in taking part in clinical trials, so that you can hear firsthand from individuals what that is like, who have experienced it or whose communities have been through that. And then finally, we'll have Viking Therapeutics, who will be providing an update on their clinical program. So with that, next slide, please. Let's start talking about clinical trials. Next slide, please. I'm going to try and go a little bit quickly, but I do want to make sure that you know that I work for a company that runs clinical trials. ICON PLC is one of the larger clinical trials companies. We don't develop any medicines. We're like a project manager for clinical trials. And that ICON is a service provider to the pharmaceutical industry, and that I am personally affected by a different rare disease through my family called hereditary hemorrhagic telangiectasia. And although my objective today is to share information about clinical trials that is general and broad and even a little bit specific to what you might find in leukodystrophies, and to make sure that's fair balanced, these things will influence my perceptions and therefore what I tell you. So I just want you to keep that in mind. What I tell you is not necessarily gospel. There are different perspectives on things. Next slide, please. So to tell you a little bit about myself, I've worked in the pharmaceutical and life sciences industry for almost 20 years. I have had the honor of working with many rare disease patient communities. And right now I work within ICON's Center for Rare Diseases, who really look after the rare disease clinical trials that the company runs. Along with my team, we're an extraordinarily passionate group of people. Many of us are actually affected by rare disorders ourselves. And I live in Canada, so I'm coming to you from the colder side of the border. Next slide, please. ICON has done a lot of rare disease clinical trials to date. We've had over 700 rare disease clinical trials, and I think that's probably more than any other CRO has done. So our folks have seen a lot. And I'm really happy to share with you a little bit about clinical trials tonight, because what we've also seen is that it is very hard to understand clinical trials. It's a little bit like a different country, because the logic is very different than anything that you live most of the time before you come across them. Next slide, please. And I like to travel, and clinical trials for me, especially, and now 20 years in, I still make this face sometimes when I come across different things in clinical trials, but it is very, very different. It has a very different logic from what we mostly encounter in our lives before a clinical trial, even in healthcare. It's kind of a space that has its own quirky logic that can make no sense at times without understanding why that is. So I'm going to try and approach this the way that I used to approach travel when I was going to a new country that I was going to explore for the first time. I always looked at the rough guides, because they kind of told you who and where and what and oriented you, and then you kind of go. So if I've done this right this evening, if I've made a difference, then you will probably come away with many more questions than you had coming in, and that's really to be expected. But I also hope that I'll be able to give you some insight into why things are the way they are in clinical trials. Next slide. So really, we're going to just talk about what clinical trials are broadly. And then I'd like to just touch on some aspects of clinical trials that you're likely to see in leukodystrophies very broadly. So not in any specific leukodystrophy, but some things that you might see common in trials in this space. And then we'll talk a little bit about what is involved in taking part in clinical trials. And that will then be followed by some firsthand experience or some experience from the community, which will really add color, because I can tell you, but it's not going to be what it feels like. And that, I think, is really important. Next slide, please. Okay, so the first thing to know about clinical trials is there's nothing basic about them. They're actually extremely complicated endeavors. So even the simple clinical trials in common conditions have a complete logic of their own. And then when you get into rare diseases, even that logic has quirks in the logic. So basic. Next slide, please. Okay, and you can hit next. So a little bit about the cast of characters that are involved in clinical research and some of the contexts that they have. Life sciences companies that develop the medications or develop the investigational treatments will generally have a role of designing that clinical trial. So designing how that trial is going to be conducted and carried out, and then funding those trials. They don't have permission to do any trial they want. They need permission from a regulator to proceed to in-human trials. And so they will take that to a regulator like the FDA or the EMA. And if the regulator feels that their study is ethical and that there's good reason for that study to take place, their molecule is promising, their data from preclinical studies that they did warrants it, they will allow that to proceed to in-human testing. However, that's a process that can take quite a while. So one of the frustrating things that communities always say is, when is the trial going to start? And the answer is, a lot of the time, companies themselves couldn't tell you for sure, because you'll see that it really depends on a lot of different stakeholders, and no one stakeholder controls every aspect of the process. Once that regulator has approved it, they're then going to approach different sites, and you can hit the next button, please. They're going to approach different sites and specialists who would potentially be taking part in a clinical trial as investigators, sometimes called primary investigators. And those individuals are going to approach their institutions and say, hey, can I do this trial here? We've got everything that we need. And their institutional review board is then going to take a look at that and review everything about that trial and think about the facility itself and decide whether that's possible, whether they feel it's ethical, and whether their facility has the equipment, has the expertise in order to run that trial. So again, there's nothing a sponsor can do about that, and there's nothing a regulator can do about that. That really will be down to each institution. Then, of course, there are the participants and medical heroes who take part in clinical trials and will take part in all of the testing. When we talk about testing in clinical trials, we're talking about testing that's taking place in patients, on patients, with patients. So it really is a medical hero role, critical. It cannot be done without individuals doing this. We cannot have clinical trials. We cannot have evidence that treatments are effective. It is the way it is that we have to pass through this space of clinical trials in order to get to the other side, to hope of a cure or hope of a treatment. And they actually hold the key to how fast you go as well, because depending on how quickly you're able to recruit individuals and how that goes, whether any of them decide to leave the study or to stay in the study, that can impact how long it takes for a study to be conducted. And then during the study, there are a separate group of individuals called the Data Safety Monitoring Boards, and they have kind of an oversight role. They will be able to take a look at the data if anything's happening and decide whether it's safe to continue. So should there be something that it was unexpected, the Data Safety Monitoring Board can come in and hit pause and say, okay, we're going to take a look at this, or they might come in and take a look at something and realize that it's going really well, and then there might be a change to the trial there. So This is the cast of characters that are broadly involved in clinical research, and they all have their own timelines and their own considerations. And it's really, clinical trials happen through the negotiation between them to try and arrive at the same common goal. Next slide, please. So fundamentally, clinical trials, and there's just no other way to put it because it's what they are, they're an experiment that's conducted on human volunteers. They're warranted. It's medical scientific research for a good purpose. We're looking to see whether a promising treatment is effective, but fundamentally it is an experiment, which means that we don't know how it's going to turn out. We have some good data that might suggest how it could turn out, and we think that based on that, it could turn out well. But nobody can give any kind of guarantee, because if we could, we wouldn't be doing the trial. We wouldn't need to do that. And the types of trials that I'm referring to and we'll talk about today are called interventional trials because they are testing something. So this is very different from a trial that is observational or that doesn't involve testing anything where you're just watching the course of the disease. That can be a natural history study or an observational study. Very, very different when you're testing an actual molecule, much, much more involved. And one of the differences between those two is the number of tests and assessments and questionnaires. And there are myriad tests, assessments, and questionnaires in a trial that's trying to prove that a drug is working or is safe and effective. And every single one of those tests will be counted in the data that is reviewed by regulators and reviewed by the FDA, the EMA, in order to decide whether that drug is safe and effective. Every single one. So prior to when we had the internet and digital things were really accessible, the way that companies would take data from clinical trials to the FDA would be they would drive it there in trucks. So you would have a convoy of trucks that would arrive at the FDA chock full of binders. So the amount of data that goes to a regulator to review from a trial is immense. It's enormous, and that takes some time. And of course, that regulator will then decide whether that is safe and effective through that review, and if so, it will provide a marketing approval. Next slide. So this particular slide, this is a pretty famous infographic. You've probably seen it before and really speaks to broad drug development. So not rare disease specific, not leukodystrophy specific, but in general, when companies try to find a treatment that is effective in a condition, this is what that looks like. So this will start years before the clinical trial phase begins. So the clinical trial is never the beginning or the end of the story. And, if you get to a clinical trial from, working with 5,000 or 6,000 or 10,000 compounds that could potentially be promising, by the time you get to clinical trials, you might be left with just a handful that are promising enough to test in humans. Everything else is going to be abandoned because in the subsequent testing, it will not have lived up to the hope and promise that it had as a molecule. So you might end up with 5 that go to clinical trials. And then in the standard model of clinical trials, that's approached in sort of 3 phases. This will be different in rare diseases, but a little bit different. But in a first in human clinical trial, which is a very important milestone, you're often looking to find out more about what dose would be effective and well tolerated and whether it's safe. So you're looking at the adverse events that are happening. More than the efficacy. So that's why in the common conditions, these trials are often in healthy volunteers, more often in healthy volunteers, really just for safety and dosing, what dose is having some sort of an effect, not necessarily effective, but an effect. And then in the second phase, which are also fairly small studies, you would generally be looking for to explore safety further. These tests might be in patients in common conditions, and then based on that, you would start to look for effect as well. But really the phase 3 trials are where the massive amount of data is generated. And in common conditions, phase 3 trials have thousands of people in them. There might be more than one that are required in order to prove that the drug is safe and effective and can last, anywhere from 3 to 5 years. So The thing I want to highlight here, and the reality of science and what we come to clinical trials with and what we get on the other side, is that for each of those 5 drugs that was promising and entered clinical trials, at the end of it, we might have 1 that lived up to its promise and proved to be safe and effective, and that would go to the FDA. So I want to point out that, unfortunately, even though clinical trials are the only road to hope, and especially in rare diseases like the ones that affect my family and your loved ones, the reality is that it is often a road that does not lead to success. And that's really important to know, that it's well worth pursuing, but more often than not, it does not work out. So with that hopeful message, next slide, please. Okay. Things are a little bit different when it comes to rare diseases. So in the standard, when I described the standard model and you saw there's hundreds and thousands of patients, that would not be possible in a rare disease trial. So thanks to the efforts of the rare disease advocacy community in the 70s and 80s, that changed. So the regulators became more flexible and allowed differences in how clinical trials are run so that they could actually be run in a rare disease patient. Imagine if they required, 2 studies with 5,000 people for every rare disease. That simply would not happen. But that's what you require for common conditions. So having studies with fewer number of participants, and having those first studies in humans actually be available to patients in some cases rather than healthy volunteers if there is urgency for it. In some cases, not in all cases, but it might be that those first in human trials will have an actual patient population. In order to save time, because time is of the essence, and this is a lengthy process. What you saw on the other slide, you're looking at decades to get from the start of a clinical trial to approval. They allow a combination of phases. So they allow companies to design clinical trials that do multiple things at once rather than making them do it one at a time. So you can combine dosing and safety and do that in a phase 1 and 2, and you can combine the last part, you could do 2 and 3 together, or aim to do that. And the great thing about that is it does save some time. It does mean that you are actually moving faster. That is never how it feels when you're on the other side of it. It will always feel extraordinarily slow, but it is done in order to move faster. The other implication of that is that participants are doing more tests. So if you're in that trial, you're going to do a lot of tests in order to address both of those. And the other difference is that there are accelerated reviews. So there are mechanisms for once clinical trials are done for regulators to prioritize those reviews for rare and orphan conditions, which are probably things that you knew already. One thing that does not change is that a regulator has to be convinced that a clinical trial, so the experiment, the way that it was designed, is going to be adequate and well controlled. And we'll talk about what that is in a little bit. But essentially, they must be convinced that if you do this study in this way, what will come out of it will be reliable and convincing to the regulator of safety and effectiveness. And if not, then a regulator is going to turn around and tell you they want you to change your study design because they're not going to be convinced by it, essentially. Next slide, please. So when I talk about study design, clinical trials have these things called protocols. And a protocol is a very, very large document with many, many pages that most participants never see. But every aspect of a clinical trial has been planned meticulously long in advance. So long in advance that it's a real pain to change. If you want to change it, you have to go back to the regulator and tell them what you want to change and then wait for that change. That's called a protocol amendment. Happens a lot in rare disease trials as well. But it's a very important document, and it has a few things that have implications for participants that are really important. So every single protocol in any disease, any rare disease or any disease will always have these elements. It'll have an eligibility criteria. So it will specify who is eligible and not eligible based on what criteria. Usually this is criteria related to the progression of the disease or tests that have been done, or what kind of other parameters that are clinical that you would need to meet in order to be in that trial. And there will be there's always going to be a disclaimer that it's always, the opinion of the investigator. So it is up to the investigator to decide as well whether you are eligible or not, whether they feel that it is a good fit for you and not dangerous for you, not too dangerous for you, and whether they feel that you fit the criteria. So it's important to know that a company will not be deciding whether or not you can be in their trial. That will actually be the call of a physician. And they can't say to a physician, put this person in the trial if the physician doesn't feel that it's safe for them, which I hope is slightly reassuring for you. Another extremely important part of a protocol, and we'll talk about this one as well, is outcomes. So a protocol will always specify the endpoints, and that is what the proof is. So the outcomes are what you hope it's going to do. And often these things are specified in the form of, gaining XY function, or having XY result, or slowing XY process, or XY progression. And that XY progression, that XY thing you're going to do, that's how you're going to measure it. So there's always something that they're going to, something that they're attempting to do, what the outcome is if it's successful. So if this drug is successful, then this person has experienced 10% slower progression. And how are you going to measure that? Well, we're going to do XYZ tests. And how they're measured is very, very important. And in rare disease clinical trials, outcomes and how they're measured can be extraordinarily difficult to get right. So companies really want to try their best to get that right. We'll talk about what that is. But that also tends to slow the process down a little bit sometimes. Every single protocol will have something called a schedule of assessments, and that is an exhaustive list of every single test that participants would have to do while they're in the trial, and in what sequence. So every single study visit or every single interaction or test is going to be laid out. And it will also specify how and when and under what circumstances the study drug would be administered. Every protocol will also tell you what the controls for the trial. So since the trial is an experiment fundamentally, the control is going to keep that experiment running without bias. And we'll have to eliminate bias, and will help to ensure that the measurements that you're seeing are accurate. So we'll talk briefly about that too. Those are actually a major source of frustration, the controls, for everyone. But they're really necessary in order to convince regulators that the data you have is actual proof. So they're very important. And very importantly, every protocol will specify what will happen if there are adverse events that are not expected. Going into a clinical trial, you always have some idea of the adverse events you might see, but never all the adverse events that you could possibly see. So there are provisions in the protocol for what you would do if you happen to see adverse events come through that were unexpected, or any adverse event, and they go back to that data safety monitoring committee if they're serious. Next slide, please. Okay, so outcomes, probably the toughest thing about a trial, because if you get these wrong, you can have a drug that's effective, but not be able to prove it, which is the most heartbreaking scenario. If you get these wrong, you might have a drug that's not effective that has potentially gotten approval and people are using it. Neither of these two scenarios is very good. So the reason why this is so challenging in rare diseases is because a lot of the time you just don't know. There isn't enough scientific knowledge about them, as there is with common conditions. That's why you have natural history studies and observational studies that often precede the actual trial. They're trying to get the information to help them to get these measures right. And I would probably describe this endeavor as being akin to trying to measure a forest, or how much a forest grew. Where would you begin? And here's a ruler. Because a lot of the measures, like the tests, one of the tests that patient communities often dislike is the 6-minute walk test. These tests are not tailor-made for the disease. Sometimes there are tests that are tailor-made for the disease, but oftentimes you're working with a toolkit that was never specifically made for that disease. So you're trying to make sure that what you measure with that will actually give you a good result. So it's a little bit like if I was writing an experiment and I was trying to measure the size of the Empire State Building, but I had a tiny ruler, or if I was trying to measure, who knows, if I was trying to measure the volume of a cube and I had 3 tools at my disposal, none of which would do the job. I don't know if they're going to do the job particularly well. So it's really important that the outcomes are well chosen and that they mean something to patients as well. So when companies come to you and ask you about your experiences of the condition, about what's important to you, they're trying to get these outcomes right. So please share that information, because as I said, it's just for rare diseases often not available in the literature the way it would be for other more common conditions. The medical tests, medical tests that are chosen will ultimately prove that outcome. So you want to get those right too. A lot of what will then happen is if you'll have a battery of tests because you're trying to make sure you don't miss anything. So for example, if you're thinking about measuring how quickly someone walks, but the condition might have any other kind of manifestations, and how I walk might only be a part of the story. Maybe I don't walk any faster, but maybe I feel better when I'm walking, or maybe I'm more energetic when I'm walking. So how you measure is really, really important to capturing that, and that companies will spend a very long time on this before being able to go to a clinical trial, and regulators will pick this out with a fine-tooth comb. So they have to be convinced by the outcome measures, and if they don't think you've picked the right measures, they will tell you no, and you'll have to go back to the drawing board and find other ways that they will agree to. Next slide, please. Next slide, please. Okay, controls. So controls have a special meaning in clinical trials. Remember, I mentioned this is a very idiosyncratic space. When we talk about control, when we talk about efficiency or efficacy, we're talking about comparing something, an intervention, to something else. Even when there isn't a different cohort, we're comparing it to something. So there always has to be something. It could be compared to another treatment, or it could be compared to no treatment at all. Or in some cases, it may be possible to create a synthetic prediction. So to take data that we have available and use that to model what might have happened and compare. Whether you can use any of these controls or not depends very much on the nature of the condition, the nature of the tools you have available to you for measures, and the nature of the drug. So these are not options that are available to all studies or to any scientific endeavor. They're just the range of controls that might be how you might approach that. So what you end up getting is you end up frequently getting 2 cohorts in a trial, right? So the people who are getting the thing, and then the people who are getting the thing that it's being compared to, whether that's for the whole trial or for part of the trial. And we usually refer to one as the treatment group and the control group. And this is, next slide, please. This is the control is usually a point that is very hard to kind of accept in a lot of times for families and for individuals, because the things that we do in order to better control the study and eliminate bias, all of them have one thing in common: they eliminate knowing. So everyone has biases, doctors have biases, all of us have biases, and if we didn't do things to prevent those biases from actually affecting the way that we perceive information, they might potentially change the way that we perceive that information. So if a doctor believes that a patient is on a drug, they might be tempted to look at the way they walk today with more optimism and think, hey, that looked a little better. And that's not what you want. You want to be able to convince the FDA that the science was good and that bias was eliminated and it still proved effective. And some of the controls that you will commonly see, although definitely not in every trial, are randomization, meaning that you will be randomly assigned to one or the other. Nobody controls that is. It will be done kind of in an automated fashion. So your doctor cannot choose to put you in one or the other. Blinding, which is really, again, blinding means that you don't know which group that you're on. And it can be a double blind, meaning your doctor also doesn't know which group you're on. The researchers don't know which group you're in. They can be triple blind, where even the people analyzing the data that's coming out of the tests that you're doing do not know which group you're on. That really depends on the circumstances of the science and the condition. And then the famous placebo, unfortunately, that's when the control group is getting no treatment at all. And fortunately or unfortunately, regulators really demand placebo control as much as possible. So if you can do it, if it is considered ethical to do it, they will generally want you to do it. Not all the time, it's not always ethical, it won't be in every trial. But it's very common, even in rare diseases, to see regulators require a placebo control period, even if it's not for the whole part of the trial and you just start and there's a placebo control period, and then if all goes well, everybody gets the drug. But it's very common to see that. And it's very hard on participants to participate in a trial not knowing what they're getting. Next one, please. Adverse events. This is something that comes up a lot. So folks are often nervous about adverse events in a trial, what is being kept from you, what could actually happen. And the first thing to know about adverse events in this quirky space that is clinical trials is that an adverse event is not the same thing as a side effect. An adverse event is technically anything unsavory that happens while you're in a clinical trial. So that's anything bad, regardless of whether it had some sort of a connection to the drug or not. So as I mentioned here, if I'm in a clinical trial, everything that's bad that happens is technically an adverse event, whether I was hit by a falling airplane part and I got a weird bruise that I wasn't sure where that came from, all of that would be reported and considered an adverse event. It may have no link whatsoever. And that will be something the data monitoring, the clinicians in the data safety monitoring board will do, the DSMB will do. So if it's something serious, let's say that falling airplane part hit me and I died and I was in a clinical trial, the DSMB would look at that and see, is there any possible way that that could have been linked with the drug? And if it's determined that it's not, they'll make a call on that. If it's determined there was some possible way that it was linked, maybe I just didn't move fast enough because it was slowing me down or something, then it'll be looked at and considered whether the trial can proceed. That's the quirky logic of trials, because we come at it with the idea that we have some understanding of what this molecule or treatment could do in the human body, but we certainly don't know for sure, especially if it's only ever been tested in very few people or not in people at all. So the safest thing to do is to assume that anything bad that happens could have been linked to that and then investigate that. So an adverse event in a trial is not the same thing as a side effect, which is really what we find out about in trials. So at the end of that trial, we will have a better idea of what actually is linked to the drug and what was just random chance, or perhaps just happened because the disease progressed the way it would have anyways. Next slide, please. Okay, so I did want to touch on some broad things that you are likely to see in clinical trials in leukodystrophies. And I do want to emphasize this is broad. This is definitely not any one trial or any trials in any specific leukodystrophy, but these are just some themes and trends that you're likely to see. Next slide, please. Okay, this is a build, so we'll go one at a time. First one, please. So clinical trials in leukodystrophies, because leukodystrophies are such complex conditions, as you would know better than anybody, are complex trials. These are complex trials. The trial design is complex. It's taken a very long time to define the outcomes. It's usually a matter of doing many tests. These are complicated and they are not the easiest trials from a participation perspective. These are generally very, very demanding trials of individuals. Next slide, next build. Okay. So we mentioned that trial designs are different in rare diseases often, and you will definitely see that in the trials that are taking place in leukodystrophies. So you're unlikely to see 3 distinct phases. You're very likely to see that 2 of those phases are being combined in some way, which adds to the complexity of the trial. The aim, again, is to try and save time, but what ends up happening is you're doing this trial over a longer period of time. So for everyone, it just feels very long, which it is, because we want those answers today. But you're very likely to see trial designs that are attempting to gain speed by doing multiple things at the same time. Next slide. Sorry, next build. I touched on this before, extensive medical testing. In general, leukodystrophies clinical trial designs involve far more testing, far more frequently than you would have in a natural history study, which is just looking at, kind of lurking in the background and taking information from your standard of care treatments. And it's definitely not the same as an observational study. A lot of testing is required in order to see what it is doing in all parts of the body and to see whether it's reaching where it's intending to reach. Next bill, please. And on that note, you're very likely to see that the compounds in these trials have a route of administration that's fairly invasive. It's going in somehow via the cerebral spinal fluid. And the reason for that is that most compounds cannot cross the blood-brain barrier. That's probably something you already know as well. And what that means is that some of those tests are going to be invasive because you're trying to find out what's happening where this is actually intended to have an effect. So unfortunately, they do tend to involve invasive blood draws and CSF draws and administration that involves accessing the CSF either through the base of the spine or the base of the head or in some way in that way. So this is some, these are very, very, very intense experiences to go through medically. Next one, please. You will also see trials in this space using a cohort approach quite commonly. And what that means is that, let's say a trial says that it's going to recruit 40 people, because in some cases, there'll be no human trials before. It'll be first time in patients. And in some cases, because you just don't have enough information about how that might affect and you want to take a more kind of a safer or more cautious approach, you're going to have a cohort approach, which means if the study is going to have 40 people, what might happen is that initially only a few of those people will be given the medication or the intervention. So you might have a cohort that starts with only a few patients. Sometimes it's even possible to have something called a sentinel patient, which is like 1 or 2 patients that will go in and then you'll see what happens. And that first cohort, what will happen is they will receive the treatment. There will be a pause and a period of monitoring what happened to them and to determine whether, if that looks fine, then you can go forward with the next cohort, the next few people in the cohort, and then you would do the same thing and proceed. Very frustrating for the patient community because often people mistakenly believe that the trial has been canceled or paused. That's not the case. This is a specific way of approaching trials in order to try to combine those things, do those multiple things at once in a cautious way. So a few patients first, and then the next, and then the next. And next one, please. Very long follow-up periods. So this is something that after you finish the clinical trial, you might still, depending on the type of trial, have quite a number of years left of follow-up. So being continually, and when I say follow-up, that refers to going back for tests and being monitored and having your health monitored for quite a while to see whether the effect is continuing. Next one, please. This is probably the most heartbreaking one, and this one is true of many rare diseases and clinical research in many rare diseases. Due to the limits of the science and to the shortcomings in our measures for outcomes, what ends up happening is that it's very common to see very narrow inclusion-exclusion criteria with, requiring people who have very similar, who are very homogeneous in terms of their course of disease, the stage of disease, their context. And that's because you're trying to eliminate potential confounding factors, and you're working with outcome measures that are only so sensitive. And the heartbreaking reality is that in many cases, clinical trials, the majority of the population in a rare disease will not actually meet that criteria. And that is, extraordinarily difficult for every single patient community that I've ever worked with in a rare disease, but it is very common. It's just a fact about trials that doesn't really get talked about as much as it should. In reality, very few people in a rare disease are likely to actually meet the criteria if you were to look at the actual population. And there are reasons for that. They are often to do with the limits of science and the limits of what regulators are willing to allow. The ALS community has done some incredible advocacy lately that led to the passing of the Act for ALS, and that attempts to address a little bit of about that for their rare disease community. But I just want to make sure that that's stated, and that you're aware that clinical trials are usually available to everybody. Next slide, please. Okay, and finally, another thing you're likely to find in leukodystrophies is different types of trials with different designs and different implications, because they're investigating different types of therapeutics. And across the leukodystrophies broadly, there's quite a range of different types of therapies that are being investigated, everything from small molecules, which might be chemical compounds, to proteins, biologics, mAbs, and so forth, to ASOs and gene therapy. And that's quite a range, and they have very different implications. If you happen to have multiple options available, if there are more than one approach that is being tried in your particular disease, then it's really vital to inform yourself about every single one in detail. Very broadly, and this will not be the same in every trial, but very broadly, there is a bit of a difference between gene therapy trials and the other trials, and it is a bit of a spectrum on the other side here. So with these other three, not with gene therapy, you're more likely to have had some data from tests in healthy volunteers. That will definitely not be the case everywhere. Some studies will absolutely go to patients first in human, but you're more likely to have data available from healthy volunteers in this side. Gene therapies will never be tested in healthy volunteers. These three trials here, the small molecules, biologics, ASOs, they are less likely to limit your opportunity to part in future trials, meaning if you did one, there's probably a chance you could do another one in the future. That is, with the caveat that usually you can only really do one trial at a time. And usually, let's say you were to drop one and want to go into the other, there would be a period during which you'd have to wash that out. So if that was permitted and you were not excluded, then there would definitely be a period of time when you would have to be certain that it's out of your system in order to take part in that trial, so that it didn't confound the results. However, with gene therapy trials, this is a one-shot deal. Once you've been in a gene therapy trial, you will not be eligible for further trials. And this is because that will be a confounding factor forever. Once you have a change to your gene, at this point, we're not able to have the science to determine whether the effect that you might be seeing in another clinical trial came from the gene that was put there years ago or not. And so the exclusion criteria for trials will usually be no gene therapy in another trial. Doses. A lot of the first studies that are going to be available in leukodystrophies and in rare diseases include dose ranging. So the ability to change doses is actually kind of an interesting one to keep in mind, because with these three here, you may be able to change doses. Let's say the dose you got or the dose you were assigned to won't be so good. In the future, you could take it up a notch, let's say potentially. Or with gene therapy, that will not be the case. So in gene therapy dose ranging studies, you'll be assigned to a dose. But that administration, depending on how that gene therapy was administered, may make it not possible to have a second dose. So for example, if a viral vector was used, your body will develop antibodies to that. And at this point, we don't have a way to scientifically overcome that. So a second administration, even of the same gene therapy, may not be possible. So that's something to keep in mind. On the upside, with gene therapies, you are not going to have a placebo control in that trial. However, in common molecules and in the other types of therapies, you'll be more likely to have a placebo control. It will not always be placebo control, but that will be more possible. And then of course, possible treatment, possible cure. We don't know either way because they're still in clinical trials. So we won't know until that trial is done. Does a gene therapy always lead to a cure? No, absolutely not. Even when it's effective, that doesn't mean it's 100% effective. That doesn't mean it doesn't leave gaps. It's certainly not that every gene therapy is going to cure or provide a cure. It might just provide a better disease course. And a possible treatment could be just as a possible treatment could be quite effective. So the thing is, you just can't tell until we're on the other side of the clinical trials. And this is one of the reasons why individuals who take part in clinical trials are absolutely medical heroes, because you are taking a risk, in the service of yourself and in the service of your community. That is one of the greatest legacies that I could imagine someone leaving. Next slide, please. I'm actually going to end here rather than giving you my thoughts on clinical trial participation, because I want to make sure that we have enough time to hear from the panelists who've experienced this firsthand. So I think you can take down the slide deck so that we can begin the panel. And I would like to welcome joining us this evening are Ms. Amy Price, who is metachromatic leukodystrophy parent advocate. Also joining us this evening are Marcy Flores, who is a parent and ULF board member, and Mrs. Rojas, who is the mom of Brandon, who was affected by ALD, and Brian, who received gene therapy in 2015. So thank you very much for joining us, Amy, Ms. Rojas, and Marcy.
Thank you. Hi, we're here.
Hi, I'm very excited to hear from you and to hear a little bit about your experience. I wonder if you could just first say a couple of words about yourself and whether you'll be addressing questions from your own experience or from the experience of your patient communities. Amy, let's start with you.
Thank you. Sorry, I'm like floating in the dark here. It's that time of day. My name's Amy Price, and I have a son who underwent gene therapy in 2011 for metachromatic leukodystrophy. I generally speak more from the community perspective and recognizing that our experience is only our experience, and it's useful to have that big picture. I also am lucky to work in a field where I work with clients, companies that are trying to elicit insights from patients to inform their drug development process. And so a lot of times I do hear very similar things, whether it's leukodystrophy or oncology or any other space. And so I will speak to my experience, but also ensuring that I'm representing others as well.
Thank you, Amy. Marcy.
Hi, I'm sorry I have my camera off. I had parent-teacher conferences today and I did it for like 3,700 students. So I'm just getting home. I apologize for not having my camera on. My son is 17. He has 4-H leukodystrophy. We've been at this for about 12 years. I've been on the board since January, and it's I'm very honored to serve the ULF community.
Thank you. And Mrs. Rojas?
Hello, everyone. Yes, so we were affected with adrenal leukodystrophy. It was passed on by me, and we didn't have any history in our family. And my son was affected at 7 years old, and Brandon, and then thanks to him, we were able to find out that my little one, Brian, also had the same diagnosis, and we were lucky and blessed to find out about the clinical trial, and we went through the process, and very, very grateful, for how Brian has been benefited from it.
Thank you. So the first question, I'd like to kick it off while everyone is typing in their questions. And we did have some questions from registration that I'll put out to you. But my first question is, can you share a little bit about the decision-making process that either yourself or that you've seen families go through when the opportunity to take part in a clinical trial becomes available? And I'll put that out to any of the three of you ladies about the decision-making process.
I can speak on our experience. It was very rapid from the point of diagnosis. Like many rare disease families, we had never heard of metastatic leukodystrophy, and our son was diagnosed because our daughter, who was older than him, was showing early signs of MLD, and finally we were able to obtain an MRI, and that's where it was revealed that she had a type of leukodystrophy, and then the additional testing to determine what type. She was almost 3 at the time. So from the point that she was diagnosed and our son was confirmed to also have MLD, then we had very short, what felt like an emergency. Like in our mind, when you find out your child has a terminal diagnosis, you almost can hear the ticking clock. And I had already conducted some research, and we were very lucky to have a doctor that was really well connected with ULF and in the leukodystrophy community. And it was 3 weeks later, we were on the ground in Milan and beginning the process for our son to go through gene therapy, and he was patient number 2. That treatment decision-making process was about, in our mind, even our daughter was, she was declining rapidly at that time, but it was about survival. I mean, it was really felt like a race against time, and it felt like weighing the pros and cons, not necessarily not treating. That didn't feel like an option at all to us, but there were bone marrow transplants or other options here in the US that reviewing outcomes didn't feel like a good choice to us. So it really was just about, this is the option and the only chance that we have. So I think that for us, that was what drove the treatment decision making. For a lot of people, there's other factors that can influence that. There might be other burdens that would make going halfway across the world and being there for 6 months and leaving your jobs and all those things are insurmountable burdens to other people. And then similarly with other trials where there might be extensive travel or additional expense or other issues, when you're looking at, I mean, this is very much a family process. It's not just about the patient, but it's about, in pediatric case, the caregivers, other children in the family, kind of the whole family unit being impacted.
Thank you, Amy. Mrs. Rojas or Marcy, do you want to share a bit about the decision-making process that you've had or that others in your community have gone through?
Hi, I can share ours. Like Amy, our son Brandon was declining very rapidly. And once we found out about Brian, we were devastated. We were told about bone marrow transplant, but there was no match for Brian. And so, and when we were told about gene therapy, and we said, that's it, that we have to do, we have to do something, and it has to be done fast. So we had a meeting with the Boston team, and they, they were very supported, and they said that we were very afraid because Brian was only 3 years old, 3.5. He was gonna be 4, and we were very scared that he was so little. But they assured us that the youngest the patient is the better. So we decided to go for gene therapy.
Thank you. Marcy?
Hi, unfortunately, with my son, by the time, first of all, he has 4-H syndrome, which is like polar 3. So we're in the early stages of research. They just identified a couple of genes not too long ago. So they are exploring, ways to have gene therapy for our population. Unfortunately, we're not there yet. But I will say this, based on the community, the overall leukodystrophy community, and those families that were given the opportunity to go to pursue a clinical trial. I would have done it myself if it was a possibility. I would definitely jump at the chance. Anything, right? Like we would scale the earth if it would cure my son, or if it would prolong his life or anything like that. So although my son is different, our situation is different, the progression of the disease and the course that it's taken, it's just not in his future. If I was given that opportunity, I would absolutely, I would take that chance and see where it would take me as a parent.
Thank you. Now, I have to apologize because I neglected to introduce other members of our panel that are able to join us tonight. So I'd like to welcome Dr. Migawa and Ali Fatemi and Maria Kefales, and to apologize for that. Please feel free to jump into the conversation at any time. And it'd be great if you took a couple of seconds to introduce yourself as well to the group.
Yeah, I'm Gustavo Migawa. I'm a biochemical geneticist at Columbia, and in pediatrics and neurology. So, I see patients with XALD as well as other neurological disorders and genetic disorders affecting the brain. Thank you. I'm happy to be here.
Thank you, Dr. Migawa. We're really thrilled that you could join us this evening. Mia?
Yeah, my name is Maria Kefales. I'm with the Calliope Joy Foundation and Cure MLD. My daughter passed away 7 months ago from MLD, but I have been very fortunate to work with MLD gene therapy families since 2013. And so we've been our community has been on the front lines with the ALD community in terms of lentiviral vector gene therapies, and we now have dozens of patients around the globe that have been treated, and we have EU approval with EMA, and we actually, which is now public, we had our first child identified through newborn screening in Germany through a pilot, and we expect that child will be treated in Germany with Limeldi, which is going to be among the first commercial uses of Limeldi in the EU and will be the first time we've used it with a newborn screening child. So this is an amazing kind of moment for our community.
Thank you, Maria. And Dr. Fatemi?
Hi there, I'm Ali Fatemi, a pediatric neurologist at Kennedy Krieger Institute. It's good to see you all.
Welcome to the panel. I wonder if Dr. Fatemi, Dr. Migawa, and Dr. Kefales, whether you could talk a little bit about the decision-making process that you've seen families or individuals approach opportunities to take part in trials with.
I can take a stab at that. So I think it's disease specific. and then once you get into each individual disease, it's really many factors that dictate it. But for the most part, I think people want to participate in trials. I think it's a highly motivated community. I think the challenge comes in when it comes to the actual logistics, operational logistics of these rare disease trials, and so it's always like the devil is in the detail, you know. So everybody wants to participate in the trial, but then we say, well, you have to be abstinent of this and that for the next 6 months, or you know, you have to come for the first 2 weeks every 2 days and stick around. That becomes, those things can become major barriers, you know, especially in working people who have, you know, who are working and have to get away from work, and in families who have young children. You know, think about a gene therapy, you know, like for instance, the Rojas, I mean, they live in New Jersey, New York, right? And then you have to go up to Boston, and then a very sick child, so it's very complicated. You know, these kind of challenges are a lot there, but I think for the most part, it's a highly motivated community to participate. But then it's really, again, disease specific. You know, one of the diseases I'm working on right now, LBSL, is a very slowly progressive sort of spastic disorder of the legs with ataxia. Cognitively, most of these kids are basically normal. And it's really a question like, you know, we're not there yet, but if we get to injecting an AAV9 virus into the brain, do they really want to take that risk or not? Because these kids are living a life, you know, they have impairments, but, you know, they have significant quality of life, and there's potential risk of their life at risk to undergo these therapies. On the other end, you have kids with, you know, adult childhood cerebral ALD, where, you know, they're too late for transplant, and we know what's going to happen to them, and you know, they will do anything to get their child to treatment. Their doctors will do anything to get those kids to treatment. So I think that it really is depending on each individual disease, and you're talking about now 50 something diseases here, so it really varies.
In the MLD community, it's been a remarkable trial design. So, you know, and Amy can speak to this more directly, but most part, the children who were diagnosed and eligible for gene therapy, the families had to diagnose an older child to save a younger one. And it was, I think, incredibly challenging for the families and the clinical team to basically say, you know, we can only help one child. And we've had families where mothers were pregnant and expecting children after the news of a MLD diagnosis. So I've been in awe of the families and their courage and bravery. We also have a Takeda enzyme replacement therapy trial, which we filled in record time, despite this fact that the requirement was 2 years weekly infusions in a port in the spine. And the Takeda team was like, how did you fill this trial so quickly? And it was really and they've stayed in the trial. They have stayed. I mean, I think as a patient advocate, it was my job to kind of serve like Laura's job, a CRO, in terms of telling families, this is a clinical trial. You don't have to do it if you don't want to. But if you do it, you must take a 2-year commitment. You must be committed to 2 years. You must be committed to doing the follow-up, because you can't waste a slot that way.
However, you are always free to leave a clinical trial.
You are.
You're not contracted to do that. You have a right to leave a clinical trial.
Oh no, they absolutely they do. But I when I had families like, so what was interesting from my patient's side, right, Laura, which I think is a really interesting question. So I had a mother who said to me, well, you know, they told us we can leave. And I said, yes, it's true, you can leave, but if you're not willing to make this 2-year commitment up front, if you're already thinking about leaving, this is not a curative therapy, it's not going to save your child, then maybe you don't have to. I gave them permission not to do it, but I also did ask people to make the commitment, because these slots were really precious. And I had learned, and you know, I'm not a PI, I'm a parent, and I'm like, I do feel very strongly that I've seen families take clinical slots and they weren't committed to the process of follow-up care. And we lost data, and we lost, and they were not good matches for clinical trials. Now, I'm not a clinician, so I'm not driven by the rules you guys are, but when I see what our families go through, I do not want people who are not committed to the process and who are not going to try. I mean, they can leave anytime they want, but if they're saying to me first week, well, I'm not sure if I really want to do this, then I say, then maybe you don't want to do this for 15 years. And it's okay. I give them permission not to. More than that, I don't want people doing trials if they're not committed to a 15-year commitment, or 2 years, two times a week. But I'm clearly I'm making people squeamish, but I feel very strongly. I really feel very strongly about that.
I think this is one of the things that is kind of unique to a rare disease, is that clinical trial, whether you're in it or not, represents hope for you. And so it's for the community. And you do have that dynamic where you know that every person who leaves is a delay. They're going to have to go back to the drawing board and get another person who starts at the beginning, and that's a delay. And then you have to balance that with, and we hear patients balancing it in a different way with, maybe it's legitimate to feel that it's no longer right for you. That can happen. There are legitimately good reasons why people can have the right to make that decision. I wanted to ask, and I think this is one for you, Amy, Mrs. Rojas, and Marcy and Maria. What does it feel like to be in a clinical trial? What does that actually look and feel like as a participant?
I have to think about that one for a moment. I did want to touch on, Maria, the point that you were just making about the not staying in a clinical trial. I think that's where my squeamishness would come from. I think that puts too much of the burden on the patient and the family. And if that's, I don't like companies having that perception, because then I think these needs around psychosocial support and the practical barriers that families face, from financial to childcare to impact on the family, are like, well, we don't really need to worry about that because we know they all want to be here because they all want to save their child. Like, I think that there needs to be an awareness that all of those needs need to be met, and it should be easy for families to want to stay in trials.
And I want to say too, Amy, that as an advocate, one of the most important things I do is fill in the holes that the clinical trial companies can't fill. So, for example, our foundation has raised hundreds and thousands of dollars for compassionate use. We have fought to get insurers to reimburse for compassionate use. When a family got stranded in Chicago because they were a Polish family and they needed to go back home, we got them back to Poland. We had a family that was stranded in Pittsburgh. We got them back to Pittsburgh. And I got into fights with clinical trial providers because they weren't advocating for those children and families. And Amy told me all sorts of things that she thought could be improved in clinical trials, from having, most famously, hospital gowns that were the size that were for children. And we found a volunteer to make the handmade gowns that are still to this day used in Milan. So, I mean, I am not an employee of the company. I am a champion for our families, but I have gotten into it with many PIs over the years.
Yeah. And I know you, I absolutely know you have. There's so many therapeutic areas that don't have a Maria. They don't have the force of that filling in those gaps. And so I think, how do we make sure that we're not just looking at other people to fill in the gaps all the time?
Agreed, Amy. That, I think that's what advocacy should be doing. And I think that's a really crucial part that I think people, they raise $5 million for the clinical trial, and then they don't keep the families in the clinical trial, and they don't support the families while they're there, and they don't help with the problems. Like the big problem we had with the international trial, which was terrifying to me every night, was the fact that we had families from Brazil and Poland in America. And if those babies got sick with pneumonia, there was no way that the parents would be bankrupted
Mhm.
if they had a hospitalization. And Takeda was like, well, we can't do anything about this. And so, because it's coercion, they couldn't pay the families. And so, we were in this really nightmarish battle to open sites in their home countries so they could go back home and get the infusion and not face $1 million medical bill for an ICU hospitalization, or that the children were going to die because they couldn't get a feeding tube. So, I mean, the role for advocacy, I don't think people fully appreciate the things that advocates have to do and families have to do to be in clinical trials. And it's a space that, PAGs fill in ways that I think a lot of folks are quite naive about until they have clinical trials running in their community.
We have a question from someone from the ALSP community, and they wrote this into their registration. Have you ever decided a clinical trial was not right for you and your family, and what was that deciding factor?
I think that it wasn't necessarily the same trial process, but as I mentioned, when our son was diagnosed, and even our daughter, she was early symptomatic for late infantile MLD when she was diagnosed. There was the availability of bone marrow transplants here in the US, but the outcomes were not what we would want as an outcome, A, for our child, and B, the process to put them through to get to an outcome that wasn't what we would hope for our child. So both of those, the outcome and the process, were, I even had somebody reach out to me and say, well, your daughter, even though she's early symptomatic late infantile, we can, she can do the bone marrow transplant. And I just thought, from my personal experience, why would I want that to be her last days? Like, I want her to be sitting on a beach and eating ice cream and laughing at Caillou. Like, I didn't want her to be in that situation. So, and it's a very difficult decision to make for every family. Other families might choose that, and I absolutely 1,000,000% understand where that decision would come from, because we're all just trying to do the best that we can for our children.
We had one family that opted out of Takeda, and the decision was interesting. They were actually really close to the site in Amsterdam, and they simply just decided it wasn't worth it to them, that they wanted to spend time eating ice cream and watching Caillou. And I think that a big part of my job was helping them be okay with that decision, because there were about 5 other families who wanted that slot and were not eligible. And so they felt tremendous kind of pressure from the community to publicly to acknowledge the decision they'd made, which was absolutely fine for them. And so, I mean, I think that, again, as an advocate, you're trying to always deal with, when you have a clinical trial, it's amazing. Dr. Vandiver once said to me at CHOP, she said, people think that when a clinical trial opens, things get easy, and that's like a big success. Actually, the community, it's very hard for the community, and it becomes a haves and have-nots. And so I think so much of advocacy becomes really managing people's expectations, talking about, providing support for families who don't get treatment, providing support for families who do, families who get really good outcomes and families who don't get great outcomes. So as a parent of an untreated child who died, I feel like I have a tremendous amount of power to say, I totally understand how you feel. I am heartbroken every single day, but at the same time, I respect the process of science and understand that not everyone can be in the trial, because trials won't work if we let everyone in. And that's very hard to explain to families who are desperate and angry and just are feeling like this is an arbitrary decision made by a scientist. And I tell them the doctors they wish they could give everything to everybody. They just can't. They just can't. I must say that once a week to people.
We wish we had a lot of money to open a lot of clinical trials to help patients. But it's usually the sponsors and the opportunities of trials that we don't get. I think Ali knows that many submissions go to NIH and sometimes other sponsors that FDA eventually turned down that would be worth trying, but sometimes I guess the money is short. for trials, and then there's limited number of patients as well that they can afford the recruitment, which is sad.
We have a question from someone in the Alexander disease community, and that's a community that has a clinical trial that's ongoing now. And this really struck a chord with me. They said, why is it so tough to get information during a clinical trial? I wonder if Dr. Fatemi and Dr. Magawa, you could take a stab at answering that one.
Again, I'm not sure what they're exactly referring to. If it's referring to obtaining interim results, if it's about interim results of the trial, there's a process that has to take place, right? So people have to get the treatment, and there is usually a drug monitoring committee that closely monitors the process, and you have to wait until you know if it's working or not, and so results are reported usually analyzed every year or so, unless there is something really noted by the investigators that stands out. Like if people start dying, you may not wait for a year. If you have a serious adverse event, you have to report it usually within 24 hours, and so that's, I guess, you don't know, because people don't know yet what these trials are showing, because they just started and they're ongoing and they're in the middle of them. If that's what they're asking. If they're asking about, whether why do we not know whether this trial is going to get expanded, or new sites being added and so on, those, specific to the company and the sponsor and the investigator sites and so on. And just one thing people need to understand is that contracts and regulatory stuff take forever. It takes easily 6 months to put together an IRB protocol and get a clinical trial agreement between two entities. And it's not that people are slow. It's just that there's usually a lot of things that need to be reviewed, a lot of things that need to be negotiated, and ethics committees come up with lots and lots of concerns and lots of questions that need to be addressed. And that usually the end product of that is a 25-page long consent form that finally everybody agrees on, that they want every patient to read. So I don't know if I'm addressing the question. Maybe Gustavo is better.
Thank you, Dr. Fatemi. Certainly, and I wonder if maybe Amy or Maria or Mrs. Rojas, you could expand upon that. What are the set types of information that patients and families want during trials, but generally don't get and find hard to deal with?
In my experience, that wasn't a challenge through our trial process, and I haven't spoken to others within the MLD community that have had that difficulty, but I have in my work spoken with others within pediatric metabolic conditions where families reported, we conducted a gene therapy roundtable, that they would join investor meetings that were being broadcast so they could hear data from their own trial, because nothing was being shared with them and their children were participating in the trial. And so there were a lot of conversations as we shared this information back to those that sponsored this roundtable around in ways that they can communicate and what's the best way to share information to these families that are traveling and staying places for a long period of time and participating in these trials, so they know when they take this child home, were they stable, what were the outcomes, giving them some type of feedback. Can that be done with communication with a doctor, maybe in their local area, rather than coming directly from the site? So just trying to ensure that families have that information. They don't have to go seek out finding it in places that are being shared with investors, but not with the families.
Mrs. Rojas, when you were and your family were going through the gene therapy trial experience, did you find it easy to get your questions answered, to find information about the trial or the information you would have wanted?
Yes, we were, my husband and I were very involved in finding out. We called a lot of people that were not able to go and do the gene therapy. We were able to talk to people that did do the gene therapy. And so it was available for us. We called the doctors a couple of times just to make sure that, you know, things were going good, and he was was eligible for gene therapy, and thank God they were very accessible. Like we were, you know, we didn't have a hard time getting an answers. And then, you know, we were very positive about it. You know, we were just, you know, hoping and praying that the gene therapy will be what our son needed, you know, for his health.
Thank you.
I'm going to be a contrarian because it's late and I'm tired. But I mean, I don't think that there are any secrets in clinical trials anymore, because people are live streaming on Instagram feeds and they have 10,000 followers on Facebook. And I mean, I was talking about a very big gene therapy trial for an LSD, and my colleague in the GM1 space, Christine Wagner, was pretty terrified because one of the families was describing, the mother was a writer, and the mother was describing in Instagram stories every single day of the procedure and describing, you know, pictures of the child's response. And, you know, I kind of joke with my industry partners in clinical trial, you know, PIs, like, there is no such thing as a blind trial. People are live streaming and giving neurofilm numbers outside. So I'm not sure. I understand, like, you know, there's official readouts that may be slow to come, but actually social media has made it, and it's dangerous and scary sometimes, the kind of information that's out there from patient families.
We sometimes hear, and it's very contextual, it really depends, but we sometimes hear frustration from patients around not knowing when the trial will start, when the site will be open. And again, that's down to that really not being something anyone actually knows for sure. You're going to find out once the site starts working on it, and find out once you're in the trial. It's not always possible to have results. It's often not possible to have results of the many, many tests you're doing, and that can be immensely frustrating for individuals. If you're seeing, I think it's different when you're seeing a massive change, or if the treatment or something is such that you're like, oh, I can actually see something, versus when you might be talking about reducing progression or slowing progression, you don't necessarily see it, or it's not tangible. So I think that can be a really different experience in terms of the information and the desire for that information to give you certainty. I'd want to just ask one final question before we turn it over to Viking. What kind of support do people who are taking part in clinical trials need? And what is generally offered? What is missing?
I think that it's key to, in preparation for trials, to ask the patient community that question, obviously. And then I think the piece that I often see as missed are really those practical and psychosocial elements of that can weaken your potential trial participant or a care partner, caregiver, if those needs aren't being met. I always will see parent, patient go into a clinical trial with all the strength in the world to battle the medical side of it, while they're crumbling inside over things that are like, getting a taxi or calling an Uber, or the little things that are often overlooked. Difficulty at an airport, not knowing where to get groceries in an area if they're in a new city, not having their social support system. Laura, going back to something you said at the beginning when you were just describing clinical trials and what are they, you said, we don't know, I even wrote it down, we don't know how it's going to turn out. That we don't know how it's going to turn out is a psychological burden that families that go through this process, especially with something like gene therapy that is still emerging, will carry every single day of not knowing how it's going to turn out. And you'll hear the doctors will still say that. I mean, my son's 12 now, and he was 12 months old when he went through trial, and every day I'm still like, if he's not feeling well or any little small thing that's completely unrelated, he's a normal 12-year-old boy, but it's that never leaves the mind. And so I know that like they couldn't address that at the very beginning, but how through the process when people are going through the clinical trial, do we look at the whole person rather than just the elements that are related to their participation and showing up for tests? What's everything else that is piling up on them?
Thank you, Amy. Mrs. Rojas and Maria, what are the kinds of support that you needed or that you see other people need in clinical trials?
We were just hoping that the clinical trial will work for Brian. We didn't have to worry about anything else. There were everything was provided for us, a place to stay, and we wanted to be together. Even though my son was declining, we kind of split our jobs with my husband. I was with my son at night with Brandon, and my husband was in the hospital with Brian while he was receiving treatment. We actually had our parents also with us. So we kind of had the whole family helping us with, with food, and, it was just, everybody was involved, like our close relatives were involved. And so that was really a support for us, to be together as a family, and to give them both all we had, our strength, our energy, everything, to be able to have a good experience, even though it was something that, like you said, that we don't know. We still, we're hoping that everything is good, but, sometimes we question, how is it gonna be in the future. But, like I said, we, with this diagnosis, we increase our faith and just, prayed every day.
Thank you. Maria, is there anything you want to add in terms of the type of support that you see people needing in clinical trials? Oh, you're muted.
I basically have listened to Amy for the last decade and taken her advice on nearly everything that I could, because she's an incredibly wise and smart person and is one of the pioneers in the field, and really a hero of mine. Like when I asked her early on, like, what did you need? And we have done everything, a lot of stuff that that she's talked about. We've really worked to implement, the psychosocial stuff, and a toaster oven and bikes for the team in Milan, because the Italians want bikes, which is funny. Like at CHOP or Boston Children's, you would never give families bikes, but the Italians wanted bikes for patient families. The liability issues, I guess, are different. But I think one of the more difficult, actually one of the most difficult incidences in my advocacy career came 2 years ago when we had our first pre-symptomatic baby that was not able to get treated. And the reason the pre-symptomatic baby didn't get treated was because their family was low income. and we could not get the family opted out of compassionate use and took a more affordable Medicaid available therapy, and that haunts me every single day. I think that the one family who didn't get this life-saving therapy was because of economic resources, and we've been able to. We had a perfect record until this family, and that is a challenge that will endure for gene therapy and clinical trials in general. That the fact that families with resources and families who can mobilize connections and networks can put pressure, raise money, get media attention, and other families are denied that life-saving opportunity.
And how that impacts that delayed diagnosis from the very beginning, the longer diagnosis being impacted by that.
There are a lot of, like you said, Amy, other things than what's actually happening in the trial that individuals have to do to be part of that trial, millions of things, and how they feel when you're in the trial is so much more intense. You often have people describe, if you're having a bad day at the airport, but when you're in a trial, it's a million times worse because you've got the added pressure of the trial. And the way that things are, different trials offer different levels of support, different funding. It's different from sponsor to sponsor to trial because there are no laws that say this is how you always have to do it. And that's a real barrier to equity in clinical research. It's one of the key barriers that a lot of individuals and advocates are working to try to change, but that is unfortunately the reality of clinical research today. With that, I'd like to thank you very much for joining the panel. And I know we're a few minutes late. Thank you very much for bearing with us, Dr. Neutel. Keely, I wonder if you could get Dr. Neutel's slides up. We're lucky to have Dr. Neutel joining us this evening in order to share an update from Viking Therapeutics on their efforts in the leukodystrophy space. And I will turn it over to Dr. Neutel to introduce himself and to take us through it.
Well, thank you very much, Lauren. It's really a great pleasure for me to have the opportunity to spend a little time with you all this evening, and I've thoroughly enjoyed the program thus far. My name is Joel Neutel. I'm a clinical professor of medicine at UC Irvine and the director of research at the Orange County Research Center in Irvine, California, and also a consultant to Viking on many of the studies that they would be doing and that I'll be sharing with you tonight. It was very nice to hear Laura's presentation because I think it gave you a lot of insight into the planning and all the thought that goes into developing new drugs and the studies that need to be performed in order to get those drugs available to the patients who need them if they do become approved. And I've been working on one such drug for a few years now, one which I think is potentially very exciting for this particular condition. And we still do have to go through the research, like you've heard, and there's a long path. But some of the early stuff that we've seen, I believe, is very exciting and gives us hope that this may become very important. So if you can give me the next slide, please. I'll give you a little bit of background of how this all came about. And I know that everyone on this call is very familiar with the fact that when we look at XALD, it's caused by a mutation in the ATP binding cassette transporter D1, or more commonly referred to as the ABCD1 gene. And the ABCD1 functions normally to transport the very long chain fatty acids into peroxisomes. And in those peroxisomes, when they get there, they are broken down and excreted out of the body. In XALD, we know too that there's a defect in this ABCD1 gene that leads to accumulation of these very long chain fatty acids in the tissues. And these are the villains in subjects with this disease, in that they cause disruption of cell membranes, they cause an inflammatory demyelination in the brain tissue, and they also cause motor neuron deterioration. Now, the drug that we're going to talk about is actually looks a little bit like the thyroid that we normally make in our body. And there's been some very interesting research surrounding thyroid and stimulating the thyroid receptors. And this turns out to be, I think, very important in this particular disease. We've known for many years that thyroid hormone is very effective in modulating the metabolism of lipids in our body. And had it not been for the fact that thyroid as a hormone, potentially if it's given in higher doses, has some negative effects, I think that we probably would have used it much more frequently in managing difficult disorders of lipid metabolism. But what's become interesting from several studies in recent years is that the thyroid receptor, as I'm going to show you some more of in a moment, is really divided into two parts, an A part and a beta part, or alpha and a beta part. And it's been shown that if you specifically can stimulate the beta part of the thyroid receptor, it can stimulate expression of compensatory transporters, ABCD2 and ABCD3, which actually may mitigate the very long-chain fatty acid elevation in patients who have XALD. Now, we've learned for many years that the body many times have alternative pathways. If one pathway is blocked, it can develop other pathways to overcome that block. And it would seem from the recent research that these other two genes in this family may be those alternative compensatory pathways which may be able to help alleviate the problem of accumulation with very long-chain fatty acids. Next, please. Now, as I said to you, the thyroid receptor is divided into two parts. They both were important, but if overstimulated, the alpha part of the receptor has some very negative effects. It can cause arrhythmias in the heart. It can cause elevations in heart rate. It can have effects on bone and cartilage. And so it's very dangerous or not good to have very high levels of thyroid hormone, which actually stimulates this part of the receptor. And like I said, that's why we haven't used it as a treatment for lipid metabolism problems. However, we also know, if you look on the left-hand side of the slide, that the beta part of this receptor is the one that regulates very acutely lipid metabolism. And if you can stimulate that part, it has a dramatic impact on lipids. And you've also seen now has an impact on stimulating these alternative pathways for the metabolism of a lipid, which is a very long-chain fatty acid. Next one, please. So here you can see, like Laura was explaining to you, you have to go through this very long process of looking at various drugs in cells, animal models, and then in normal human subjects in order to assess the safety and to try and assess the efficacy of these new agents. And you can see from the slide that when the gene the gene for adrenoleukodystrophy related protein is regulated by the beta part of the receptor. And when you stimulate this, and these are from cells, fibroblasts of patients with this disease, you can see that there are several drugs that actually stimulate and increase the formation or the development of these alternative genes, which may be very important as alternative pathways. You can see from the slide that this particular drug that we're studying, which is VK0214, is very impressive in the stimulation of this beta part of the receptor, which results in this overexpression of ABCD2, which may be very important in correcting the buildup of long-chain or very long-chain fatty acids in these subjects. And you can see those, it's really more significant in terms of that expression than we've seen with many other drugs. Next one, please. Here you can see the same thing looking at the expression of ABCD2 in the various tissues that were in no way important in patients with this condition, namely the liver and the brain. And you can see that compared to placebo, and Laura told you about placebo, you can see that gene expression in the cells treated with this particular drug, which is called right now VK0241. Oh, I'm sorry, 0214. Next, please. Here you can see the first animal model that was used with this particular agent. And we know that the best animal model that we have to mimic what we see in patients with XALD is the so-called ABCD1 knockout mice model. And in this model, these animals are treated and modified in such a way that they really have a problem with the ABCD1 gene, and therefore it mimics the biochemical features that we see in human subjects with XALD. And here you can see that when these mice were treated with this particular drug, VK0214, you saw progressive reductions in the plasma very long-chain fatty acids seen in these animals. And you can see from these slides that at 25 weeks, there were fairly impressive reductions in C26. These are all the very long-chain fatty acids, 24, 22, and 20. And that occurred in the blood, in the plasma. And also when we looked in the tissues, you can see that the same thing occurred in the tissues, that there were significant reductions in the liver, in the spinal cord, in the brain, in those very long-chain fatty acids in the mice treated with this particular drug. And this was very encouraging data to show that this lipid metabolism and the stimulation of or overexpression of ABCD2 by this agent really did seem to have a beneficial effect on the very long-chain fatty acids in the best model we have to mimic what we see in our human subjects. And again, like Laura said to you, these are the studies you want to do first and the tests that you want to do to assess that there really is a path forward to take these particular agents into human subjects. So following this, we did exactly that. Next, please. I have, like I said, a research unit, a large research unit in California. And the first studies that you do with these newer agents, like you heard, is in healthy volunteers. And so we started in a fairly good size, and I've now studied several hundred healthy volunteers who have been given this drug, looking, firstly, is it safe to give to human patients? Are we going to see any unexpected side effects that we did not see in animals? And is it going to give us a signal that will do the things that we hope it will do and that we saw in some of the preclinical studies? And here you can see we took healthy subjects who had very marginally elevated LDLs or cholesterol levels. And you can see that at doses of 25 milligrams and more, it started to have fairly significant decreases in low-density lipoprotein cholesterol in these healthy subjects who started out with levels that weren't so high to begin with. And that was very encouraging for us because it proved that it really does have an important impact on lipid metabolism. Next one, please. We looked at other lipids in these patients as well. Another one that's very important as a marker, as a sign for lipid metabolism is triglycerides, another one of the fats that we try and manage in subjects who have it too high. And again, you can see very significant reductions in triglycerides in healthy volunteers who started out with levels that were not that elevated to begin with. And you can see reductions up to almost 50%, which is very impressive and is very hard to achieve with any of the medications that we have available thus far. Next one, please. We then went and looked at some of the more complicated lipids, ones that we really don't have very much in the way of treatment for these lipids. We know that they're important in terms of causing disease. We just don't as yet have good treatments for them. And you can see when we looked at things like ApoB and Lp(a), both of which are known to be very important in the genesis of cardiovascular disease, there was in small groups of patients very nice trends showing that there's a reduction in these lipids that we haven't even had drugs to treat up until now. Again, emphasizing very clearly that that part of the receptor is very important in terms of lipid metabolism, and we can impact it by stimulating it, and we do it specifically without having a negative effect on the alpha part of the receptor. And so in these patients, we didn't have any side effects of the cardiovascular system and on bone and cartilage, because it was shown to be very specific only for that beta part of the receptor. Next, please. With these same subjects, again, healthy volunteers who started out with normal long-chain fatty acids, very long-chain fatty acids, you can see from this slide, and again, we only looked at small numbers of patients, but again, very impressive trend demonstrating in people who didn't have high, very long-chain fatty acids to start out with, that there is a reduction even in these subjects of those long-chain fatty acids. And we've had some discussions with Dr. Fatima as to the importance and how you can extrapolate what we see in normal subjects to what we may see in patients with SALD. So these are things that are important, exciting to see, and clearly suggest that we need to look at this a little bit further. Next, please. Now we're running out of time, so I'll go quick so that there'll be some time for questions and answers. Here you can see again the very long chain fatty acids in the same group of subjects, and the same exact picture, this trend to showing reductions in subjects who started out with normal values in the beginning. Next one, please. So, and here C26, same picture. So all the various long chain fatty acids we looked at in the study, and same picture again. Next, please. You might say, well, are these people going to get a problem with their thyroid? I mean, we're stimulating a part of the thyroid receptor. Are they going to have thyroid abnormalities? Are they going to have problems with their thyroid hormone in their body, which we don't need under normal circumstances? And we were able to show that there really was no significant change at all, and none of them had any problem at all. In fact, it was very well tolerated. And we've seen that when you use a more physiologic way to manage these sorts of things like lipid metabolism, it really is not usually associated with a lot in terms of side effects. Next, please. So in summary, from what we've seen, the data was very encouraging in terms of safety and tolerability. There were no serious adverse events seen. And like I said, I think clearly showed for us and for the FDA that we were able to isolate just that beta part of the receptor and not have any impact on the alpha part. And as a consequence, there was no evidence of cardiovascular effect. There was no significant impact on thyroid and the thyroid axis, and there really there was no safety signals at all that we were concerned about. And as a consequence, now the plan is, and is currently ongoing, to proceed with looking at these doses that you've now studied in healthy volunteers in patients with XALD. And we're going to look at the 20, 40, and 60 milligram dose. Next slide, please. And the ongoing, because of all these promising effects in terms of very long chain fatty acids, and because it was so well tolerated. Next, please. It's a once-a-day tablet that you take in the morning. So there's no infusions, no invasion at all, just a tablet that's taken once a day in the morning. And this is a study that's currently ongoing here in the United States at many sites across the country, and also now in several sites in Europe, where we look at patients, males with AMN, and doing what we call a parallel study. You heard that from Laura, where we're comparing placebo to various doses of this particular agent and treating the subjects for a month and measuring all the things that are important to assess whether in fact they are having an impact on the very long chain fatty acids and other markers that we can see in this study. And so this is ongoing, I think very exciting, and hopefully we're going to see very good results from this. And the hope is to get the study enrolled as quickly as possible and then have that data so we can take it to the point where it can be approved, if it works well, for subjects with this condition. I think, Laura, I'll stop at this point just so we have a little time for any questions, and I know it's nearly at the top of the hour, and I'm sure people have to get to their evening. So forget about the last slide. Let's go on. You're on mute, Laura.
So thank you very much for your talk, Dr. Nuttall. It's wonderful to hear that. We had some questions from the community that came through the registration. I wonder if you would be able to share anything about the kinds of individuals who the trial would be open to, what types of patients that would be open to most likely?
So yes, sure. So like I said, we try to keep it as broad as possible for patients with this condition. It's going to be in men with in males with clearly diagnosed genetically and through very long chain fatty acids who have this condition. And really, if they fulfill those inclusion/exclusion criteria, can go into the study. So we've tried to keep it as broad as possible. Again, for reasons like you said, you know, we want to try and include as many as we possibly can right now.
Thank you. Let's see, we've got another question here, which is there a possibility for bone marrow transplant individuals? I'm actually not sure if that's coming from the same community, but is there a possibility for individuals who've had a bone marrow transplant to join the trial?
I believe so. I don't think that there was an exclusion criteria for the study. So yes, I think that they can.
Thank you very much. I'm trying to look for other questions from the LSP community. There is one question that asks, what is being done to connect with the adult community? I wonder if you can speak a little bit about what, oh, we got a question here from Luann, sorry, let's go to that one. Can men with both AMN and ALD be in the trial?
Yes.
Okay, that's a great answer. And what do you know anything about the ways in which Viking is trying to connect with the community? I think your presence here tonight is an obvious example of trying to connect with the community and inform them. How is Viking trying to connect with the adult community of both symptomatic and asymptomatic people?
So I think, like you say, through communications like this, to help describe and get people to see what's ongoing in terms of the current studies. And then we have a number of excellent clinicians and people, many of whom on this call. Dr. Fatimi is one we work with very closely, and others who we are going to be working with on these studies, and you know, have done so much for this community and are working so closely with this community, and hopefully through their good work, the word can be, you know, disseminated out to the patients who have this and the opportunities made available.
Thank you. We have one last question, which is a classic question. Approximately when will the study begin enrolling?
It has already started.
We are enrolling
as we speak.
We are enrolling.
Yeah. Dr. Fatimi is enrolling already right now, yeah.
Have them call me. My email is everywhere.
We have another question, whether a 15-year-old male with advanced ALD, would they be considered for the trial?
I think it might be a good question. Do you know the answer to that, Dr. Fatimi? I think they have to be 18 or older.
18 or older.
Yeah.
18
yeah.
or older.
And Dr. McGaw as well. There's another one who's working on this. So thank you.
Thank you very much, Dr. Nuttall, Dr. Miguel, Dr. Fatemi. Thank you also to Ms. Rojas, Amy Price, and to Marcy and Maria. Really appreciate, and we appreciate every single person who's watching this evening to take this back to their communities, or who's viewing this recording later. Thank you to each and every one of you for everything that you do to advance research and to advance towards cures and treatments in rare diseases. This is your life's work, and I want to recognize and acknowledge that. It is always something that gives me a lot of hope and courage to see individuals continuing to do that. So thank you very much for this evening. I will now turn it over to Bob.
Thank you so much.
All right. Thank you, Laura. Especially thanks for leading this webinar and obviously the discussion to help everyone learn more about clinical trials in the leukodystrophy space. I also have to thank my ULF staff for all their behind the scenes work. So thanks, Keely and Tara. Next, I really want to thank all the panelists that took their time, because I know their time is so valuable. And so we as ULF, and I know as patients that are participating in this, we appreciate it. So special thank you to Dr. Fatemi, Dr. Magua, Dr. Nuttall, and our great panelists, patient panelists, Lillian Rojas, Amy Price, Maria Cafalas, and Marcy. Your help has just been great to be willing to share stories and information to everyone that's on this webinar today. So I know our families appreciate your coming here tonight to help us learn more about the clinical trials that are taking place and the challenges faced by the families that have participated in them. If people want more information about ongoing clinical trials, you can go to clinicaltrials.gov for more information on your specific leukodystrophy. Lastly, I want to thank the families that have attended tonight, and I hope you've learned something about clinical trials from a patient perspective. I'm wishing all of you the best as you work your way through the complications of life with leukodystrophy. So again, thank you everyone who's attended tonight, and this webinar will be available on YouTube in the next couple of days. So have a great night, and thanks for attending. Appreciate it.
Good night, everyone.